From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. Episode number 2, recorded on August 8th, 2023. I'm Mark Martin, your host, and welcome to today's Quality Quorum. This is our second episode. For our initial segment, I will discuss some shock and awe concepts about the microbial world that I introduced to my microbiology students. After that, I will discuss 3 important questions about microbiology and microbiologists with my guest, Dr. Mya Breitbart of the University of South Florida. I truly hope you enjoy this exploration of the microbial world. Thanks so much for joining us today, Micronauts. And I'd like to talk about what we're going to do today. For the first 15 minutes, I'm going to be talking a little bit about how I really get students here at the University of Puget Sound, but elsewhere, interested in the microbial world. In other words, my goal, as you can see in the image here, is to teach others to see through microbial eyes. In addition, I am wearing 2 lapel pins today, naturally a streak plate. I call them inspirational lapel pins, and also a pin that I had made that speaks against the use of jargon. I find jargon often exclusionary. Yes, it's precise, but I find it keeps people from seeing the wonders of microbiology and science in general. If you look behind me, and rather than seeing the back of a wall, you can see part of my office that I've turned into something like a podcast studio. I spend most of my time in my little research lab with my research students because I really enjoy working at the bench. But we all thought it would be a better place to broadcast from here, which I'm really happy about. The Germans have a particular name for a hobby. And in the 19th century, many Germans had what was called a Wunderkamer, which was a cabinet of curiosities. And you can see some of mine behind me. And at the beginning of every one of my podcasts, I'd like to talk about something. And as you can see now, there's a flat piece of rock that's been carved with a quote from Pliny the Elder. And I won't attempt to butcher Latin, but what it actually means is nature is to be found nowhere more than in her smallest creatures. The truth of the matter is, Pliny was probably referring to things like bees. But as for me, it certainly sounds like microbes are the way to go. So how do I introduce this shock and awe concept to my students? The first thing that I need to tell you is that unlike my own experiences at large PhD granting institutions, there's only one microbiology course here. And without being judgmental, there's not a lot of microbiology being taught in the lower divisions. I do my best, but there's a lot of information that needs to be there. So one thing that I'm always interested in is what students think when they first come into a classroom. And one way to do that is to use word clouds. You ask for a student to respond in one word to a particular prompt. I use the prompt bacteria. And when I do this approach, with my first-year students, where I have a relatively large class size, you can see that you get a variety of interesting responses that sort of promote a human-centric and sort of negative way of looking at the microbial world. I also do this with my upper division microbiology students. It's a much smaller class, and I have about 16 usually. My introductory class has about 48. So for my upper division class, when I do the same thing, you'll notice if you look at the word cloud that will be up in a moment, that again, you don't really see the depth and breadth of microbiology the way I would like folks to see it. That is to say, microbiocentricity. So what I try to do is I try to shock them a little bit. And today I'd like to talk about 5 basic ways in which I do that. And what you're going to notice is there are going to be links in the show notes that you can have a look at to learn more about each of these topics. I like to hit students where they live, things that they're familiar with, and show them how important microbes are to that whole experience. There is a chemical that many bacteria make called geosmin. And the fact of the matter is, this chemical, this compound, smells like potting soil. So, one of my favorite experiences that I hope to show you in a linked video here is what happens when I have students smell a streak plate of Streptomyces. What you'll notice is that I've taught them a mantra that dirt doesn't smell like dirt, dirt smells like Streptomyces. And the first thing I'd like to tell you about that is the term dirt is often frowned upon, especially by wonderful thinkers like Dr. Joe Handelsman at the University of Wisconsin-Madison. Soil is a better word, but you actually know what's being said here. In addition, there's evidence that that geosmin compound is an attractant for small meiofonda like springtails that actually were able to attract those springtails to the areas the bacteria are colonizing and therefore transport them to other locations. So this is a wonderful first kind of shock and awe thing that I like to do with students because everyone is familiar with the smell of soil. But it turns out that what you're really smelling is the breath of bacteria. The second thing that I'd like to tell you is something that I do as a demonstration in front of them. And I start with an interesting concept. It's a trick question, though. What is the freezing point of pure water? And everyone loves to tell me it's 0 degrees C. The trick, though, is pure. Ice is a crystal, and like all of the crystal-making sets that everyone had as children, there needs to be a seed. So, in fact, ice requires a seed to form. And it turns out that many microbes take advantage of this. Now, again, often there's jargon involved when we talk about genes and proteins in bacteria, but it's interesting to note that this is one of the names of a protein that is very clear what it really is. It's called ice nucleation bacteria. So these are bacteria that put on their surface a small protein that looks like an ice crystal. So when you have water that's below 0 degrees C, like maybe -2, -3, it doesn't actually form ice until it gets some kind of seed. And as you're going to see in the accompanying video, when you put a drop of such a bacterium into this super chilled water, the effect is quite interesting, and so are the student responses. It turns out that the organism that I use is Pseudomonas syringae. I learned this technique from Dr. Joe Handelsman at the University of Wisconsin-Madison, and it's a wonderful thought piece for students about preconceptions and about how to think about the microbial world. But you might ask yourself, why does Pseudomonas do this? The fact is fascinating. It turns out that most of us know that when things freeze, the water inside them expands. And this can cause damage to what's frozen. Frost damage is a very big deal in agriculture, and it turns out that these microbes are doing this to essentially damage the plants that allow a small amount of their sap, their xylem and phloem, to leave that they, the bacteria, could then use for lunch, which is a wonderful, wonderful process. Now, I love talking about this with students, but then I blow their mind a little bit more by saying there's a lot of evidence that, in fact, this ice nucleation protein made by a number of bacteria is involved with the formation of snow. I love the idea of a snowflake having a dancing microbe at the center of it, and that's actually fairly close. Any of you who enjoy skiing, especially in areas where they have to create the snow, are using something that is called snowmax, and that's simply the freeze-dried bacteria that contains the ice nucleation proteins. What this in turn means is that many of you have been skiing the microbial slopes, We have a lot of skiers in my classes, and I love telling them about that. Finally, my guest today, Mya Breitbart, did something funny on social media. If you look carefully at this, you can see that she took an image a student of mine made with the horrifyingly familiar to me vanilla ice, to kind of give away my background. And then Mya wrote some lyrics about it that are really quite fun. No, I won't perform it. So that takes us to two of those shock and awe comments. What about a third? One of my favorite microbes to tell students about is sometimes called Conan the bacterium. Its real name is Deinococcus radiodurans. And what Deinococcus does that's so fascinating is it can survive extremely large levels of radiation, extremely high levels. And I often talk about it in my class. Where was this organism isolated? Several places. Not surprisingly, inside nuclear reactors. Also, in food that had been treated with radiation to preserve it. It is a fascinating organism. There's even some evidence that it can survive exposure to vacuum on the International Space Station. It's a remarkable organism, one of the most extreme of extremophiles. And in the image that you see here is something that I love to show students in class and then ask them what's happening. If you look carefully at the picture of a slide that I teach with, you'll see that the DNA, the chromosome of Deinococcus, is large on the gel that you're looking at. It is shattered to tiny fragments by an enormous dose of radiation. And I mean physically damaged, double-stranded breaks, torn up. Yet, 2 hours after that exposure, you can see it reassembling. And by 24 hours, it's indistinguishable from before exposure. This is a fascinating organism and worthy of an entire segment if anyone's interested in hearing more about that. It's certainly one of the students' favorite topics for writing a term paper, or as I call them, microbiographies. So the last two that I'd like to talk about with you are a lot of fun. I often get students interested in plants, and then they will tell me that they want to know more about microbiology in plants. And there's a lot to talk about, like root nodulation, crown gall disease, a number of things of that nature. But something that's beautiful beyond words are, and I have to say this carefully so I don't butcher it, the pink pigmented facultative methylotrophs. These are pink bacteria. They're beautiful. And what happens is they can live on one carbon compounds like methanol and also formaldehyde. Where are these from? Plant metabolism. Just as we breathe out CO2, there are small amounts of these one carbon compounds that the plants are exhaling. And because the surface of the leaf is so crowded with competitors, any organism that, quote, learns or evolves more accurately to use something that others can't has an advantage. And that's what's happened here. So what I love to have students do is take nutrient media where the only carbon source is methanol, and you print the leaf onto the surface of the plate. And after a couple of days, you get an outline of the leaf in these very beautiful pink bacteria. Here's a photograph of a student project where this took place with some clover, as you can see. However, if you look in the literature, you can see fabulous jobs of people who are much more precise than I could ever be. You may be asking yourself, what on earth are these organisms using the methanol for? What's the big deal to all this? Well, it turns out that the organisms, the methylobacters that are responsible for this, many of them have symbiotic relationships with plants, even making plant hormones. So it's a fascinating way to hook students into the relationship between plants and bacteria. There's even some evidence that one particular species is responsible for the smell of strawberries. And I saved the best, in my opinion, for last here. Anyone who's looked at my social media presence knows that I certainly love something that I call Lux Art. Bacterial bioluminescence is fascinating to me and has been since the time I was a little boy. I believe I mentioned that in our last podcast. But I have to tell you, if you look now, here is a culture that I started just yesterday and took a photograph of. And you can see how really bright this photobacterium is in culture. And many folks, not just me, had thought, what about painting with bacteria onto the surface of nutrient media? Can you make the equivalent of art? Many scientists have done this, including Alexander Fleming. The American Society for Microbiology even has auger art competitions. I've been doing Lux Art for many years. And what I really wanted to tell you is what fun students can have. Here are some visiting high school students that are showing you a little bit of Game of Thrones when they put their chins on some of these glowing plates. And as you can see, it's beautiful in the dark. And what I want to show you now with this beautiful photograph taken by its own light of a mushroom, students can do remarkable work. Now, I won't lie to you, sometimes people that have, for example, my skills can only manage to write a couple of words with a paintbrush. Because I remind you, what you're doing is dipping a paintbrush in the growth medium that has bacteria growing in it, and then you're painting it on the surface of a nutrient plate. And I want to point out that many people know that my wife, Dr. Jennifer Quinn, is quite the artist. But it didn't start that way. And I tell students this to remind them of the excellent work almost anyone can do if they practice. It's a good life lesson. As a mathematician, of course, Jenny loves Albert Einstein, with whom she shares a birthday. And you can see the initial attempt she did several years ago is recognizable as great Albert himself. But, you could see how most people could do that. However, after practice, look at this second photograph, and I need a better word than photograph. Well, just call it Lux Art from now. Again, imagine me in the dark room with a camera taking these photos. But look how beautiful this is. And this has been remarkable. This is a great way to promote student creativity, show how important microbes are to the environment and to everyday life. And I have, in fact, put on a number of what I call Lux Art sessions at other institutions over the last few years. If you look in the show notes, there are some simple videos showing the wonderful art that people ranging from high school students to undergraduates to professors have done in the past. And I hope someday to do a session with professional artists present, because that would be remarkable. So I like to really use this approach of creativity in my classes. So what we're looking at here, I've given you 5 different ways that I take students the first couple of days of class and show them how microbes are doing things they never had expected. And this is the thinnest tip of an enormous microbial iceberg of the wonders of the microbial world. So with that, I'm going to now move to the next segment, which will involve Mya Breitbart. Welcome back, Micronauts. It is an absolute pleasure to have our visitor and interviewee today. This would be Dr. Mya Breitbart, a friend, a colleague, and a truly remarkable scientist. How are you, Mya?
I'm great. Thanks for having me. It's a tough act to follow Vincent as your first guest.
Sure, if you say so, but I've known you for a long time. So would you mind giving everybody, I put a link to your laboratory and professional website in the show notes, but go ahead and tell the listeners a little bit about yourself.
My name's Mya Breitbart. I grew up in New Jersey. I is a child of 2 scientists, so I maybe had that direction already going. When I was growing up, I was really obsessed with marine science as a kid and didn't discover microbiology till college, which I'll tell you more about later. I am now a professor at the University of South Florida in St. Petersburg, Florida.
So, Mya, when did we meet?
Ooh. I knew you from Twitter well before we ever met. We first met at an ASM meeting, but I don't remember the
Oh, we
year.
don't talk about years
Um,
anymore, Mya.
but New
I
Orleans,
think
right?
so. And you were standing up for people in a way that made me really proud to know you, because I consider you not simply to be a friend, a colleague, a scientist. You're also an advocate, and you will stop at
I try.
nothing to defend the people you believe in, and that's really to be admired. Well, I don't want to turn this into kind of a like fest, which is kind of what it is, and this is really why I do the podcast. But I did want to ask first, did you have any comments about what I was talking about earlier when I was shocking and awing students?
I love it. I'm shocked and awed, and I learn new things. And now I want to go make
Oh,
bacterial
they're
leaf
great.
prints and smell soil. Um, and I was expecting to hear your water bottle buddies
That's later.
in there. So just for the listeners to know, there's more coming. Mark's got a lot more fun ways of getting people excited about
So may I tell
microbiology.
you, Mya, that I didn't read the papers about pink pigmented methyl facultated methylotrophs. That's fun to say. Very carefully when I was first trying this, because I like to shop these things before I expose the students to them. I shouldn't use the word expose, I guess. Anyway,
Probably not.
what I'm trying to tell you about is that all of the one carbon molecules are essentially exhaled through the stomata. And there are a lot of leaves that don't have stomata on their upper, only on their lower to conserve water. And if you were Professor Martin, not thinking carefully, and you print the upper level of a leaf all over everything, you don't get very much. So I learned that the hard way.
Hey, these are important lessons. I'm sure your
Oh, they
students learn
do.
too.
Mya, I want to say one more thing, which is in your website, and I would like you to tell people about it, because this is one of the reasons that you're a hashtag microbial hero of mine. What's the slogan of your lab?
The slogan of my lab is "Focus is for losers." And I hope no one takes that in the wrong way in thinking that if they know one topic very deeply that I think they're a loser. That's not true at all. But I am completely unable to pick just one thing to work on because I'm super excited about
Now, I
everything.
can't blame you for that. That's how I've always felt. So I'm delighted to have met a fellow microbial traveler, as it will. So
Absolutely.
I'm going to go ahead and get to the big three questions, if that's okay with you. Yeah.
Sure.
So, what single event or experience turned you, Mya Breitbart, into a microbiologist?
This is an easy one for me because I feel like, unlike a lot of other people who were interested in micro from the start, I was not. And in fact, in high school, my mom, who actually worked as a science aide in my high school, always reminds me that I was quoted as saying, "If I couldn't see it, I didn't care about it." Terrifying in retrospect, right? But I wanted to be a marine biologist, like so many kids out there. Had no interest in micro, but then when I was a sophomore in college, I took my general microbiology class, as we all do, and they did the very classic identify your unknowns experiment or lab. So in this class, students are assigned some unknown bacteria. The teacher knows what they are. We don't know what they are. And you run through this whole slew of tests to try to identify what your bacteria are. And there was something about this, about the kind of detective nature of the game that absolutely captivated me. And I loved that I could take these unknown bacteria and actually sleuth out what they were. And that was the first time that I started thinking, microbiology could be cool. But as it often is, my teaching assistant for that lab was a graduate student. And he was working with these amazing bacteria that they had isolated from sponges that were collected from under the ice in Antarctica. And they were looking for novel antibiotics and other antimicrobial compounds in these. And so I started talking to him more about this. I somehow weaseled my way into volunteering in that lab. And then all of a sudden, I was actually starting to characterize real unknowns because these were bacteria we didn't know what they were. And I'm probably going to show my age here, but that was before we just sequenced things to get the answer. And so I spent a lot of time trying to uncover what these unknowns were, what antibiotics they might produce. And I think most importantly, I realized for the very first time that there were microbes in the oceans, which wasn't something I was ever taught in a marine biology class. And that I could kind of combine these loves, this love of marine science that I've had since I was a little girl, with this newfound interest in microbiology. And I was hooked 100% from there.
Well, I'm really delighted to hear that story, because actually, I've always been interested in marine bacteria, and it didn't ever occur to me that I could make a career of that directly. So that's always a lot of fun. So, what type of microbiology do you study, and why does it excite you? Here we go, folks.
I love viruses. Maybe not a surprise to those of you who know me or who can see the cool phage in the background that actually Mark gave to me. Um, I So as much as I didn't know about bacteria in the oceans, even once I was working on bacteria in the oceans as an undergrad researcher, I didn't know there were viruses in the oceans. And I applied for an NSF REU experience, which is research experience for undergraduates. I would encourage any undergrads who are listening to this to look into these programs. You get paid to go somewhere and do research. And I did my first REU experience with Farouk Azam at Scripps Institution of Oceanography. And Farouk is a giant
Yes.
in the field of marine microbiology and an amazing person. And when I went there, I really had no idea what I was doing or what I was going to do. And Farouk brought me into the lab the first day and to all of his grad students and postdocs said, "Hey, here's our undergrad, somebody work with her," essentially. And it quickly turned, and no, it turned out no one knew I was coming. He had not given them any advance notice, just here is this undergrad. And so I got to work a little bit with a bunch of different people. And one of those people was Forrest Rower, who eventually was my PhD advisor. He was a postdoc in the lab at the time, and was starting to work on marine viruses. And I was fascinated from the very start because I learned how many viruses are out there, which is about, I'm going to drop all these facts on you because I love these. So, in a milliliter of seawater from surface ocean, pretty much anywhere, you have about 10 million viruses. That's about 10 times as many bacteria that are there. In the oceans, in 1 liter of seawater, there are more viruses than there are humans on the planet. There are more viruses in the ocean than there are sand grains on Earth. So if you think of anything that you think of like, well, this is the most abundant thing I can think of, there are way more viruses. And the last one, which is cool, especially if you like space and think about that as the vast unknown. If you took all of the viruses in the ocean and you line them up side by side, they would extend for more than 12 trillion round trips from Earth to Mars. So there are a ton of viruses out there. And that said, we know really well, we're learning a lot every day, but we know very little about them. And at the time when I started this as an undergrad and got into it, I was part of helping to sequence the genome of the one of the first marine phage to ever be sequenced. So a phage is a virus that infects bacteria. And before you think I was some sort of undergrad savant, I will tell you that on the first day working on this project, Forrest gave me a kit and told me to extract DNA from something. And I'd never actually used those types of pipette men before. And so I set them all wrong because I didn't realize where the decimal places were. I completely ruined the entire thing. And I was immediately hooked and was like, I want to do more of this. So I got to be involved in that, which was sequencing a single marine phage genome, but one of the first, which was very exciting. And then one of the big problems with identifying viruses is that they're really diverse. So there are DNA viruses, there are RNA viruses, single stranded, double stranded. There's no single gene that's found in all viruses, which is different from bacteria, from animals, from plants. And so we really had to develop some new ways of looking at what types of viruses are in any environment. And we did this through metagenomics, although we didn't even call it metagenomics back then because that term wasn't out there yet. Now we call this viromics. And the idea is purifying the viruses out of any given sample and then shotgun sequencing their DNA or RNA. And when this is what I worked on as a graduate student. I started in the marine realm because I've already told you I was kind of obsessed with marine science. And then I like to tell everyone that Forrest pulled a bait and switch on me, which he kind of did. And one day I found myself working on individual fecal samples, and thought, how the heck did I get into this? But maybe that fits in with the Focus is for Losers logo, because I was looking at viral communities in absolutely everything. And so we've done seawater, we've done freshwater, we've done plants, we've done animals, we've done the built environment, indoor air, we've looked at extreme environments. And the cool thing about this method is it gives you an overview of what's there. So not just one particular thing that you might go looking for, or not. So, for example, when we started working in human feces, always a fun story. Hopefully, nobody's eating while they're listening to this. We thought we're going to find human viruses,
Nope.
right? Nope. I mean, we did find some human viruses. We found a lot of bacteriophage because there's tons of bacteria in our guts. But the most abundant virus that we found was a plant virus. And this was, in particular, a virus that infects peppers called pepper mild mottle virus, or PMMOV. And we were able to track it back and figure out that it's coming from our diet. And so even if you don't think you eat a lot of peppers or spicy foods, there's a lot of it in our diets. And so we found this virus, and it turns out to be a great indicator of human fecal pollution, for example, in the oceans. And although I never would have seen this coming before the COVID pandemic happened, it's now being used really widely as a standard, as a control. So if you want to measure the amount of SARS-CoV-2 in wastewater, you want to compare to something that you know is present in wastewater. And this pepper virus has turned out to be extremely useful for that. So that's the side that I really love, is the discovery, finding new viruses in new places and in new organisms.
I can't agree more. I mean, I've always liked what I call undomesticated microbes. Sometimes they're harder to work with and frustrating because of that, but there's nothing like something new with regard to this. And there's just so much there.
My PhD advisor always used to say, if it was easy, someone would have done it
that's
already.
really true. And it's a very good way to look at it. I do want to say one thing before the third question, and I'm curious what you think of this. I think that we're very narcissistic as a culture, and I'm not. It sounds all critical because it kind of is, but people think that bacteria are bad because they only think about the ones that interact with us in a negative way. Viruses the same way. I am particularly interested in viruses that either do something positive or that we could care less about. And it's okay to be concerned about the ones that cause us trouble, sure. But I think that's a tiny minority compared to the majority.
I agree. You called them the juvenile
Yeah.
delinquents previously, and I think that's true. And actually, that was going to be one of my answers to question number
Oh.
3, was that that I want people to know that not all viruses are bad. In fact, that it's only a few of them. And I feel like public perceptions actually shifted some for bacteria, right? Now everybody knows about
Mhm.
probiotics or prebiotics and feeding your microbiome, and that having a microbiome is a good thing,
Right.
right? People seek out yogurt and other cultured products. So people have come around to this with bacteria, that they're not all bad. Viruses get a much worse kind of rep. And one of the things our lab has focused on is teaching people that not all viruses are bad. And in fact, that some of them can be good. We actually developed an activity that we call Phage Heroes, where we showed how phage, by infecting specific bacteria, for including bacterial pathogens, the bad guys, can actually be used to help us, right? And a lot more viruses just probably have no effect or no detrimental effect on their host. And we see this all the time, anywhere pretty much that we go looking. We can take a sample, you can grind it up, you can find a lot of new viruses, and these samples are animals, plants, whatever, they're healthy looking, right? Now, that doesn't mean it's not doing anything. I don't know, if I looked at a little copepod or something, would I know if it wasn't feeling well today, if it had a slight sniffle? But these are viruses that are out there in the long term. They're certainly don't appear to be causing any damage. And especially in plant virology, there's a lot of evidence that some of them can be helpful, for example, in drought resistance or in thermal tolerance. And so some of these viruses that maybe are just, I guess we would say, just hanging out right now, maybe they're going to be the thing that provides an advantage to a certain organism as climate
No,
change
I couldn't
occurs.
agree more. I think it's something that I've spent my life trying to teach about. You know, you've heard me talk about, you know, the angel microbes, devil microbes, and meh microbes, which I'm gonna talk about in a future podcast. But I think this is exactly the case. And I don't know whether I'm so excited because you agree with me or because I just think it's great science. How about both?
I like
So
it.
that is the third question, what you would like the public to know about microbiology and your research. And you've answered it, but I would like you to tell people who are deeply intriguable, if I may use that term, the range of organisms with which you have worked currently in your laboratory group to look at viruses. Check this out, folks.
I was going to say, you name it, we've worked on it. Yeah, so there's so much attention given to human viruses, like we've spoken about before, but also to viruses that infect animals and plants of commercial importance. So, for example, with plants, right? We know a lot about agricultural crop pests, things that cause people to lose money, whether that's in agriculture, in aquaculture, in any of these systems. So, we know a lot about viruses in very few species. And then there's a ton of species we know nothing about viruses in. And so we've set out to do surveys of just about everything. So all sorts of invertebrates, things like plankton that are out there. We're working on finding viruses in harmful algal bloom species as a possible biocontrol measure. I've worked on lots of marine species, including sea turtles, which were my love as a child. So being able to look at a virus that causes tumors on sea turtles has been very cool for me. Sponges, corals. We have a big seagrass project right now. Even things that aren't marine at all, kind of in my backyard. So we did a big study on spiny-backed orb weavers, which are very cool looking spiders. That my sampling site was my backyard, literally. And I kind of love that aspect about it, right? You can send your students or yourself absolutely anywhere, pick up anything, and you're going to probably find viruses in it. And so to me that's really cool. You don't have to go to some exotic location to find a new virus, right? You can swab your desk, your keyboard, whatever. You can look
You're a professor.
anywhere, soil, sediment. Yeah, exactly. Swab your professor. That's a good
No, no, I
challenge.
don't
We'll
want to be
see.
swabbed. But yeah, and I believe you've done some work with manatees as well.
We've done a little bit, not
Okay.
viruses in manatees. So I just like to take pictures of manatees. So they're all over my walls. But yeah, lots of different organisms, lots of different creatures, and sometimes when we go looking for viruses, we don't find viruses. We find other things as well. And this again fits in with the focuses for losers. But I got involved in a project last year where we were looking at a sea urchin die off in the Caribbean. And, ostensibly my role was to see if we could find viruses involved, but we use these unbiased methods that show us anything that's there and found that there was a ciliate, so a single celled organism that is a whole lot bigger than a virus. It could almost be considered a macrobe in my world. And now all of a sudden I work on ciliates. So I think that, for me, also as things I would like people to know is that there is so much that we don't know. I think there's this sentiment in the public and even amongst scientists that all the big questions have been answered already and that there's not big exciting findings to be had out there. And I would push back against that 100%. There are so many questions. We are learning new things all the time. New technologies are becoming available that are letting us learn new things. And our view and knowledge, especially of the microbial system, but I think just of the world in general, is constantly changing. And that's a good thing. That is why we are scientists, right? To answer those questions. So I think sometimes people hear, well, you thought it was this, but it turned out to be this. That's bad. That means the science didn't work. That's completely the opposite. That means the science did exactly what it was supposed to do. And we're learning new things every single day.
Every day. And it's important to remember, students hate this, but it's absolutely true. I don't know isn't a bad phrase, it's an opportunity. And that's the reason that I love micro the way that I do, to be honest with you.
And often the answer is nobody
Yeah.
knows, right? Which I think is job security if you're a scientist, right? There's a lot we
I
don't
like it
know.
when Vincent Racaniello and I were talking about the possible models of viral evolution. You know, was it a simplified cell, a complicated virus? Was it a transposable element? And I said yes, because that's correct, right?
Yeah.
So we've talked quite a while, and it's been really exciting for me. Do you have a last minute advice for people?
Ooh. I think my advice is to just keep your mind
Yeah.
open. Like you never know what cool opportunity or thing is going to come along. But I do have one thing I want to share with you, which I don't know if you know. So this might be fun. So one of my favorite quotes that I've read in a paper, and I use this all the time when I'm teaching, and I'll apologize to the macrobiologists out there. So this was in a Nature Reviews microbiology paper in 2006, which coincidentally is when I started my faculty position. So it's an important year to me. And it's by Tom Curtis, who's at Newcastle University. And he wrote in there, I make no apologies for putting microorganisms on a pedestal above all other living things. For if the last blue whale choked to death on the last panda, it would be disastrous, but not the end of the world. But if we accidentally poisoned the last two species of ammonia oxidizers, that would be another matter. It could be happening now and we wouldn't even know. And this has just always stuck with me. And I thought you would like it
Oh,
and your listeners would like it as well. You know, we get asked sometimes, like, what is happening to the microbial communities? Are they changing? Like, are species going extinct? We don't know. Like, and these are really important parts of our biosphere. So I think that's just a great quote. I love it.
Maya, you're going to have to send that reference to me, and I'm deeply disappointed. There's not enough room on me for the tattoo, because that's a really fabulous quote. I want to thank you for it.
It's a good one.
I
Well, thanks to Tom Curtis.
Well, thanks to you, Mya Breitbart, for spending some time chatting with me about your views on science. And I want to say I look forward to having you on the podcast again sometime. I would love to sit down and talk about viruses of viruses.
Ooh, absolutely.
Yeah,
They are
I know.
everywhere.
And I'm not even pushing you about looking at some of my systems for viruses. See, there it is. So, folks, even though focus is for losers, there are limits. There's only so much Maya to go around. So please keep that in mind.
And of course, you know, it's not me, but it is my fabulous, fabulous team of students and other people that I'm really lucky to work with. And yeah, way more ideas than time or
And
money.
isn't that wonderful when you think about it?
Absolutely. Thank you for having
Thank you
me.
so much, Maya. This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department at the University of Puget Sound in Tacoma, Washington. Mya Breitbart is at the University of South Florida. Many thanks to Julia Russo for superb editing and Reber Clark for the fun music. I hope you've enjoyed being part of our quality quorum today. See you next week on Matters Microbial.